So, hello everyone again. Hi, I'm Oliver. Yeah, I call myself Microbehunter here, and it's the traditional greeting every Saturday. Hello around the world again, and I'm gonna start off with some bad news. Look at this here. Yeah, my slime mold. I was not able to revive it. I'm gonna tell you a little bit more about it. I think it died. I don't know yet exactly, but I'll talk a little bit about that. But I did, of course, prepare a couple of other things to put under the microscope today because I already was worried that there might be some problems. Yeah, and my email inbox is full again. I just realized, not realized, but somebody just told me, thank you for the information. Yep, sometimes people try to contact me and then I realize too late that the inbox is full. Sometimes I don't receive any warning. But what I'm going to do right now is I want to say hello around the world, from Austria, from North Carolina, the United States, from Vietnam, from the Netherlands, the United States, the United States, United States, UK, England, Canada, okay, the Netherlands. Yeah, it's always very nice to see that there's so many people around the world like microscopy. And so what I have here is, is a slime mold, right? And I made some several months ago. I think it was in January of this year. I started, putting it under the microscope and growing it. Yeah, and basically, when the slime mold dries, you're able to revive it by adding a little bit of water again. And yeah, today I added some water, and yeah, what you see is problem, right? So I'll explain a little bit of what we are able to see here. What you see over here, that's oats, that's some food, and this yellow thing that you see here, that is the slime mold. And normally when you add water, it should start to go back to life again. It should start to crawl around the Petri dish. Yeah, the problem that I have now is it lost its yellow color, and that's a bad sign. Right. So what I'm going to do now is I'm going to put it under my stereo microscope so that you see a little bit of what it actually looks like. I think what I have to do is maybe I have to get another one, right? But I'm going to wait a little bit with that, maybe another 24 hours or so. And if nothing happens, I'll have to throw it out and I'm going to have to start again from scratch. But for those of you that don't know what slime molds are, they are pretty cool little creatures. Okay, so a stereo microscope first. And basically, they are eukaryotes, and they're single cells, and they're actually not molds, so they're not fungi. Here under the stereo microscope, you can see here this yellow spot here. This is the so-called should be the so-called sclerotium, which is a dry form of the slime mold, which basically is able to withstand periods of dryness. And several hours ago, I added water and nothing happened, but it's yellow, right? And then over here, I hope that this one over here is going to start working. You see those little branching out, these veins, so to say, right? Basically, that's the slime mold trying to look for food. And what I have over here is I put some oats over here. So that is some cereal, right? What I hope that is that this will serve as a food source. And normally, within a few minutes, you could already see that the slime mold starts to engulf and eat up the oats. But nothing like this has happened. And the slime mold also lost color. You see, it's pretty much whitish, maybe slightly yellowish. Maybe that's not a good sign. It's supposed to be very yellow. And here it looks pretty bad, right? So I don't know. It looks pretty ill. I hope that it's actually able to survive. So I'm gonna give it a couple of more hours. Later on, we can still try to put it under the microscope for sure. This basically means that I don't think that we're going to see a lot. That is the old cereal, which acted as a food source. Again, here, the dry form, which essentially did not go back to life because it should start moving around and crawling around and trying to search for food. That's a little bit of a pity here, right? Ever smaller Jesse, thank you very much. You're always doing some research here in the background. Slime molds, of course, they are not fungi. So there actually are single cells that kind of move around in amoeboid motion, so kind of like amoeba. And what they do is if they will divide and they will reproduce, and then what they will do is they will start communicating with each other over chemical signals, and they will start behaving almost like one organism, right? Crawling around and kind of spreading around and searching for food. And that basically is one of those nice biological examples where you have a transition from single-celled organisms to multicellular organisms. Yeah, so that's one of those interesting things here. So that's why they're basically being studied. Okay, so could it be too much moisture? I was very careful not to add too much moisture. Indeed, too much water can be a problem. And I'm reading the comments now, but I took care that this is not the case. So yeah, you have to be a little bit careful in rehydrating it. Yeah. So let's. I'm just gonna go through here again. Yeah, I'm gonna clear my inbox. I'm reading the comments right now. Okay, so hello from Poland, the UK, from Slovenia. Okay, yeah, from Jerusalem. Yeah, from Belgium, Athens in Greece, Australia. Yeah, so from those LA, from Los Angeles. Yeah, so that's a little bit the problem that I'm having here. If you kind of wonder where I got the slime mold from, I'll be honest with you. I just bought it. Yeah, when you buy it, you can online. What you're gonna get is you're gonna get an envelope with a dry piece of paper, and basically on this dry piece of paper there's this yellow slime mold, and what you have to do is you have to add a little bit of moisture and water, and then yeah, you place later on, you place a cereal grain, where is it again, next to it. But what I found is just be careful, don't give it a couple of hours for it to regenerate first before you add some food. Otherwise bacteria are going to start overgrowing everything, and then that's killing off the slime mold as well, right? So this is a little bit my crisis that I'm having, because I actually wanted to show this to you and play around with it because it's really fascinating. If you really cannot wait to see some kind of a slime mold, well, in this channel I did put yeah, there are some I made videos. Okay, so you're able to find yeah, if not in this channel, then also in the previous channel, yeah, that were uploaded videos in the Microbehunter main channel. You can also find some videos. Yeah. Did you ever look at the microscopic needles on the surface of the stinging nettle? And the answer is yes, I did. Under the stereo microscope, and you can actually see how those stinging needles, yeah, they actually break off. Yeah. Every now and then, if you have a question or comment, please write it here in the comments. What I'm gonna do, however, is maybe I'm going to focus more on those comments later on. Okay, so what I'm going to do now, nevertheless, is I'm going to try to put it under the microscope in any case, simply for the sake of putting it under the microscope. Yeah, because this is a microscopy channel. However, please don't be disappointed. Okay, don't be disappointed because yeah. Some time ago I did find I could see individual moving cells, but sometimes depending yeah, on the stage of the life cycle I did not. So I have to admit I do not really know what to expect. Okay, so what I'm gonna do now is I'm going to simply add a little bit of water in here. Yeah, and I've done this some time ago, and there wasn't really not a lot to see. Okay, so no high expectations, please. Yeah. Have an idea to have a webcam streaming the movements of a slime mold. Slime cam. That is an interesting one. Thank you for the recommendation. Like, I don't know, like some kind of a live stream type of thing where 24 hours, 24/7 slime cam. That's an idea. Yeah, but the thing is even though they move, it's much nicer and more interesting to see actually on the time lapse. So what I'm going to do is I'm going to try to take a little bit from here. Okay, I wonder if I'm able to scratch something off here. Don't know. Look, it starts to flake off. It does not have the correct consistency. Honestly, not good. Yeah, it starts to flake off. That is really bad. Bad sign that it might actually have died. I'm gonna use some tweezers, because when it starts to go flaky like this, it's not slimy anymore. So that's a problem. You don't see it, yeah. So that's the one over here. I'm going to add a little bit of water anyway now. I'm gonna make a slide just to look at it. Not good, okay? So no high expectations, please. Okay, no high expectations. Maybe I'm able to dissolve it a little bit to break some cells off. I don't know if this is gonna work or not. But generally, if it starts to turn flaky, not good. Okay, so this is already a sign that I've killed it. inadvertently. A cover glass goes on top. Yeah, it's also pretty thick. Yeah, let's have a look now under the microscope. And where is the? It's sorry, it always takes me some time to find the. This is the wrong microscope. Here we go. Some bubbles. Where is it? Way too dark. But we're going to go up a little bit. And the interesting thing about slime molds is that they behave like one cell, right? So when they fuse together, then basically they become like a multinucleated cell. So first there's individual cells, and then they kind of fuse together. But the interesting thing is that over here, we actually see that there's still individual cells. Yeah. But this is, you see, no movement. Okay? No movement at all. So this is, I think, dead. Huh. Yeah. Well, at least, yeah. If you look carefully, you are able to see some cells here. Yeah, but when it's actually in this plasmodium stage, then it's kind of, how do you say, they're all fused together. Yeah. There is another, I'll jump over the questions for now, okay, because I'd like to answer them better. So there is an off-topic question here as well. So that is unfortunately a pity. Yeah. So what I'm gonna do now is I'm gonna take now a sample from the other one, from the slimy part, and then maybe we're able to see a little bit more there. Okay, so that is the one that's not from the yellow part here, flaky and dry, even though I added water, so this goes away. And we now have to make another one. By the way, in case you're wondering, I did prepare a couple of other things also to look at today. We're going to have a look at a little mini terrarium. I've got, I prepared some of the wood from 6 months ago. From a tree that has been in glycerin for half a year. I've got a rotten tomato lying around. I've got some water fleas. I've got some pollen over here. So there are plenty of things that we can look at today in any case, right? So I hope we won't feel bored. But actually, the slime mold thing was actually the thing I wanted to do. And you know what I'm going to do now is I'm going to actually now grab a sample from under the stereo microscope, because it's easier for me to actually find it. So where is this? So here that's still, and I'm gonna take some of this slimy part over here, right? Because that seems to be, I don't know, maybe hopefully alive. Yeah. So let's have a look if we're able to grab something over here. Yeah, it's slimy, that's good. Yeah, there's something on here, right? You see on the tip of the toothpick? Yeah. Yeah. And again, this should be very brightly yellow. This should be very brightly yellow, but somehow when it's ill, the slime mold, it loses its pigment. So let's give it a try. Okay, so let's go back to the desk view here. And let's suspend it in a little bit of water. In case you don't know, I've already got a little bit age-related vision problems, so actually I need reading glasses. Maybe suspend it a little bit. I mean, it's still a blob of slime. But even like this, maybe we're able to see some of the cells breaking off. I don't know what to expect. Some time ago when I got it and when I put it under the microscope, I was able to see the individual cells actually move around. Okay, so we're gonna, why don't you wear glasses when streaming? You're gonna hate my answer, because I forgot them over in the other room. So, but I'm fine. I'm fine. So, the scope view. So here we go again. Here we go. Look at this. Okay, so that is now the. We're still able to see the individual cells here. Yeah, I tend to forget them every week. So this is basically now from the slimy part, and let's go up yet more. Yeah, and it's kind of interesting what we see here is because we're still able to see somewhat, it looks a little bit clustered. Once I put it under the microscope, when you, yeah, when it was this, when it was all moving around and you could not see the individual cells. Yeah, but you're still able to see the cells here. Yeah. So yeah, let's go up a little bit. Still trouble to see. I will tell you the following, because we talk about vision and microscopes. Luckily, it's not so bad with me, but some people have those floaters in their eyes. I don't know if you know, yeah, it looks like stringy and weird things. These are actually proteins inside in the vitreous humor inside the eye, in this, yeah, and they're casting a shadow. And some people actually have such big problems with those that they actually are not able to see properly the things when they look through a microscope. Yeah, because all of the floaters are actually become even more strongly visible when you look through the eyepieces, and then they actually, even though they should not have any vision problems, actually those floaters are disturbing all the whole. Yeah, so that's. I also have a few of them, but luckily not so bad. But I read in the forum once that some folks actually have problems using the microscope, because of that. Yeah. So that is that's the thing here. Yeah, not happy looking. Okay, not happy looking, and honestly, slime mold is a disappointment. That's what I can say. It's not slimy at all, no movement, and pretty sad looking. Look, when the whole thing in the living slime mold, you can actually see the things moving around, the cell organelles floating around. Hey, come on, nothing's here. I mean, this is horrible, right? So this basically means I killed my slime mold. Okay, yeah. So that's the thing here. Yeah. Yeah. If only electron microscopes were cheap. They are cheaper nowadays than they used to be, apparently, and some of them are significantly smaller than they used to be. However, there is a slight problem with electron microscopes, according to what I've heard. What I know is the problem with the electron microscopes is you got to prepare the sample, and it takes I mean, there's this one YouTube channel, I forgot the name, and he's also doing YouTube videos using an electron microscope. And this basically, coating the sample, you have to coat the sample. All of these things can actually be done quite easily. So a lot has happened, and it actually fits on a desk, but still quite expensive. Yeah. Yeah. So YouTube glasses add 30% more to the scientist's appearance. Makes you look more. If I put on glasses, I look more intellectual. I mean, I do have my other glasses here, but the problem is I'm wearing contacts right now, right? So if I put on these glasses, I won't be able to see anything at all. So I have to take my contacts out. So. But with contact lenses, I do see better. I actually once put also my contact lenses under the microscope, and you're also able to see a lot of dirt. Yeah. So that's a little bit of a pity. But what I want to do is I want to move on a little bit before I show you some other things. Because today I also tested out a little piece of wood, and I'm gonna for those of you who've been watching the streams and videos a couple of months ago, this is a piece of oak wood, extremely hard, and I had it now in glycerin for, I don't know, several months now, I think since June or July, and I frequently also exchanged the glycerin to soften the wood. So this is from an oak tree, and some people say, it's from the oldest oak tree in Europe, over 1,000 years old. It's in Austria, right? And in Bad Blumau, it's called, for those of you who speak German. Yeah, near Fürstenfeld. Yeah, there is in the city of Fürstenfeld. Just for those of you who joined in. And from Austria, and basically they have a very old oak tree there, and I did not obviously you don't damage the tree, right? But I found luckily some branches on the ground, and this is where I took some of it along. And I tried to now microtome it. And if you now try to cut the wood directly with a sharp razor blade, which I've got here, it's not going to work because it's too hard. Yeah. So and what I've done now is the following: is I left it now longer in glycerin, and I just want to show you for those. I mean, again, some of you might know it already, but this is my microtome that I'm using. And I put a piece of oak wood in here on the side, and I 3D printed some of those specimen holders. So you see over here, I'm able to put in larger stems of flowers and plants, right? Those holes in the middle are simply there so that it's easier for me to pull it out. Again, that's the only reason. And I've got different sizes here. And what I've got here is on the side, yeah, I've got difficult to see because there's a very little color contrast. Yeah, I put the piece of the oak tree in glycerin, and I put it right next to the wall of the microtome, so that for support. And by turning this here down here, I should be able to clamp it on the table, but it's not possible now. Yeah, you turn it a little bit, and you're extending it forward, and then you're able to hopefully cut across it. Yeah. And then you're able to shave off a very fine piece of the oak tree. And honestly, this is difficult. Yeah. So I was kind of hoping that by having it, no, spilled it. I was kind of hoping that by having it submerged in glycerin for such a long time, it's gonna go easier, but actually not. Yeah. You gotta always be careful. Don't hold your thumb here. You're gonna cut yourself, right? Yeah, but it is possible to kind of shave off some of those. And let's put it on so now I put this already under the microscope over here. So I've got some samples here. And I did not put a cover glass on it yet. But what you see here is with water. And we're going to have a look at this. And this actually shows the advantages and also limitations of those microtomes. The advantages are it's quite easy to make a cut. The limitation is the cut is not always very thin. So that's a little bit the issue. So I'm going to just show you. Yeah, so this is actually one of the thinner ones. Yeah. So I'm using a regular razor blade now, and unfortunately not complete, but yeah, we're able to see that at least here some of the cell layers are quite thin. Yeah. Okay. Yeah, there's a comment here about snowflake impressions. I'm going to answer this later a little bit. Yeah, it's possible, quite easily possible to make snowflake impressions. Like always, you need patience, but it does work. I'll talk a little bit about later on when, usually after 1 hour, I'll address all of the so-called off-topic questions in bulk. Yeah, so you basically see a little bit that how this looks like, and you see that the wood is quite pigmented, a little bit out of focus also because it's fairly thick. Yeah, so this is, a little bit the thing here. We can move on here to the next one. Yeah, and you see I have to refocus all the time again. Let's go down with the magnification a bit. Yeah, I mean the patterns are quite nice, but as you see, even half a year of glycerin treatment did not make the wood sufficiently soft so that I can make very thin cuts. That's kind of my take-home message that I kind of found out. So I had when I did my first cuts after several days of glycerin treatment, it worked pretty much the same way. So leaving it much longer in glycerin doesn't really change a lot. So that's a little bit the thing that I kind of found out. And here, if with trial and error, luckily, if you're lucky, you're able to get thinner cross sections. But then you see that it's not the complete branch that's cut. Yeah. Let's try the following. Let's try dark field. This actually works. Yeah, it's dark field. Not so good because no cover glass. Yeah, this is better actually. Yeah. So this is and you see it's ripped up apart a little bit. Concerning cutting, if you want to make very thin cuts, you don't always need a microtome. You can also try to make use just a sharp razor blade, and if the tissue is sufficiently soft, you can also make careful hand cuts. So I want to show you a knife that I bought. It's a this one over here is also a shaving knife with exchangeable blades, and they're really sharp, obviously. Yeah. However, with those, I had the following problem, and that's why I'm now again using the regular blades. Is that because the distance here is quite how do you say, short. If I want to cut something, then the knife becomes thick very quickly, and then the actual knife, the edge here starts to kind of block, and go into the specimen. Yeah. So this is a little bit the problem, that I'm not able, for example, when cutting thick stems, I'm not able, to cut it through, and then the part of the stem will actually go all the way up here, and I'm not able to get a good cut because it's simply too thick here, right? So with a thin razor blade, it's actually better. I also 3D printed this here, or you can try to do something else to make it less flexible. Otherwise, it's going to bend, right? And then it's not going to cut straight again. So I've been using kind of this here, and for making those cross sections, Can cutting speed have a positive effect? Yes, you have to. The idea is the following. Ideally, you cut it diagonally like this. But the problem what I have is generally with specimens is that the specimens themselves are a little bit flexible, and as you cut, sometimes you stretch them or compress them, and this causes the thickness to become uneven. So, for example, when I extended it now, and it's still not able to catch it because it's still too deep, because from the previous cut it was pulled out a little bit, right? So that is a little bit the problem that we're having. Now here we go again. So this is, and then I put it into water, right? So that's a little bit the thing that I just wanted to kind of also show you, because I've been I kind of left this here in the glycerin, which is spilled now. So, and for taking it out, let's do this as well. You go in here. And now you can actually see a little bit better. How in here. See? It's in here. And what I've also done, not in this case, but I also 3D printed it. There's a tiny hole going all the way through, so that it can push through a needle, to hold it also in place. So this is basically this old piece of wood, which is now softer, but not, in my view, not soft enough. So maybe boiling it or so should do the trick. And now, because it's spilled some of it, I'm just going to put it back. And then later on, I'm going to add a little bit more glycerin, and maybe going to put it even into a container, so that I can store it for a longer time, or I'm going to put some more pieces of wood in there. But what I also read is that you should probably put it into glycerin and also boil it, because this will make the wood also significantly softer. Okay, so what I'm going to do is I'm going to quickly spend a minute or two, on some of the questions. The off-topic questions I'll answer later. Okay, a nice hold for the razor. I'm just going to show you what I've got here. This here is I downloaded this. It's also 3D printed, for a razor blade. But I'm going to show you this here. I'm going to take it apart, because this actually works surprisingly well. And so let's take it apart. And just a second. It's falling out. Okay. So that's how it looks like. And this here not only gives it a certain angle, but the most important thing is that it gives it otherwise it's too flexible, and then it's impossible to properly cut it, and it's also dangerous. I don't want to cut myself. So this is basically Yeah, you have to be a little bit careful here on the back, the screws, that they don't, yeah, they're a little bit deeper in, so that the metal does not scratch. Yeah, yeah. And so that's just wanted to share this with you. Yeah, this is, as I mentioned, I 3D printed this here, but there are of course possibilities maybe to use a piece of metal, or yeah, to kind of clamp it in there. So there are many possibilities, or you try to get one like this, with the problem that the blade that appears here is a little bit the distance is a little bit too short for larger cross sections. So let me quickly also read a little more here. Yeah, I'm quickly just scanning through it. Yeah, if you have any specific questions for me, please write at Microbehunter or so. Then it's or at Microbehunter Microscopy. Then it's highlighted in red, and I can see it much faster. Okay, so I'm just occasionally checking the time. With those blades, you have to be super careful, otherwise you're gonna start bleeding. So let me do a little bit of cleanup work. And then let's have a look at some of the, let's have a look at the, how do you call this mini terrarium. So I'm going to put also the slime mold, yeah, back. If you're kind of wondering, this is coffee filter, yeah. So, but yeah, unfortunately, as we just said, I think this one is not gonna come back to life, but who knows? Who knows? I'm just gonna leave it maybe for another 24 hours. Yeah? Teflon coated razor blades. Honestly, I don't know. Maybe there are. I have no idea. Yeah. So yeah, the only thing that I recommend is that if you want to make cuts, then my suggestion is obviously always use a fresh razor blade, because this actually does make a difference. So let's put this away again here, and this is all a little bit. Look at this. Glycerin is interesting. It's very oily. It appears oily, but actually it's very water soluble. So yeah, by adding a little bit of water, I'm able to remove it easily. And yeah, I think we're ready to go again. Yeah. So I'm just occasionally reading again. So yeah, let me show you a little bit how the mini terrarium is doing, which I made several weeks ago, also in a live stream. Yeah, so I have not added water since, okay, but I always kept the lid on top. Let's go in a little bit here, and let's turn on the light as well. Can you turn on the light? Okay, yeah. So yeah, so the idea was a little bit to have a source of things to look at under the microscope. Yeah, when you feel bored and you just want to do a little bit of observation. So I added, of course, some moss. What the brown thing that you see here, that is a decomposing piece of wood, which contained quite a lot of lichens. Yeah, a couple of weeks ago now, it looks pretty decomposed and brown. Yeah, the moss, yeah, it looks kind of okay, I would say, right? Yeah, I want to simply have a look at what we can find here now on this decomposing wood. Lots of nematodes, I can tell you already, right? And then maybe I'm going to take out a little bit of the moss, and then let's see if I'm able to get some tardigrades out again. Okay, so in other words, this is today a little bit of a test on whether organisms have properly survived here. I do have to tell you another thing. Last week or so, I opened the lid and out came a couple of insects. I couldn't catch them, but maybe tiny flies or I don't know what. So evidently there were some larvae, insect larvae in there as well. Yeah. So how do I keep it? On topic question, you're going to be surprised. Nothing special. Of course, it's a glass jar. Yeah, I put the lid on top simply to prevent it from drying out, and I put it next to my window at room temperature. That's it. Yeah, and right now it's winter time, so next to the window it's a little bit colder than in other parts of the room maybe, but basically room temperature. Yeah, so nothing. How do you say? Yeah, specific here. So let's do a little bit of yeah, observation again. And what I usually do is the following. You take some water. Put some water here. Didn't even center it properly. Okay, and decomposing wood is a nice source of interesting stuff. So what I do is I'm just going to do the following. Let me take some other tweezers. You take it out, simply because it's easier to reach. Look at this. Of course, there where the wood was, it's less light, and so the moss also doesn't look quite as nice. We're just going to grab a little bit here of this surface. And maybe from here as well, I don't know. Okay, rest goes back. I need more water. Of course, this is way too much. Okay, so I'm gonna show you now a little how do you say, a little trick on how to deal with that. Okay, of course I cannot put a cover glass on top. Let me turn off the light off here. Of course I cannot put a cover glass on top because it's too thick, right? So so one possibility would, of course, is you take a tweezer and you try to remove everything, right? You just pick it up, right, and you remove it, right, and put it back. And that's of course a possibility. I'm gonna show you, that's the easy way, but I'm gonna show you another one that I've seen somebody do, and I was pretty successful with that as well. And you take a cover glass. So essentially there is no right or wrong way to do it. You kind of push everything to the side, yeah? Then you go like this and you drop the cover glass on it. Yeah? Still, you gotta be a little careful with that because the microscope objective, you might still touch it with a microscope objective. So what I'm gonna be doing is for safety reasons, I'm still gonna take it away. It can be reused. This still, I'm quite sure there's pretty much interesting stuff left in here as well. And you see that, so let's have a look at this here. So I am going to, let's have another look at the scope. That's the one from before. Let's take it away. Let's take it away. I'm going to go away from let's put this away here. It's of course way too bright. Okay, so it's my 4x objective. I have to flip out the condenser. The color, rainbow colors are because of the filters I have. So this is regular bright field. Yeah, and this is how it looks like, right? You see there's some stuff moving around. Actually, I'm a little bit disappointed in the other sample that I had. There were tons of nematodes. Maybe it depends also a little bit from where it took the sample. Oh, here's one. Yeah. So it doesn't matter. We're going to go up with the magnification. Yeah. And I was actually hoping to also find some amoeba, and I'm quite sure there must be some present, but because of their slow movement, they might be a little bit difficult to find. Yeah. So you see that, nematode worms, ciliates, interestingly, not so many rotifers. Yeah. Yeah, here more nematodes. This one over here looks a little bit crooked and a little bit ill. So we go up yet more. Yeah, and this is basically some exploration that you can do, and then you can simply look around and hope to find something interesting. I personally think that this was an interesting one. Was this here? Look at this. I have no idea what this is. On the bottom. Yeah, it used to contract quite quickly. This looks. Let's go up. We had more. It seems to be obviously a single-celled organism. You can see ciliate most likely. Yep. Wow, look at this. Ah, you always discover some weird stuff. Look how it's able to contract. Wonder why it does that, huh? Yeah. So that's basically what microscopy is a little bit all about. You always kind of explore stuff, and then you discover occasionally discover some weird stuff as well. Yeah, I'm gonna do the following. I'm gonna quite tap the slide a little bit. Some type of vorticella, maybe a stalkless vorticella. I don't know. Let's tap. No, normally, no. Normally, if you tap a little bit, some organisms respond to tapping, but this one not. It's also, I think it's also fairly small. I'm using them a 60 times, so yeah, objective. So let's have a look at a few other things here. Yeah. Let's have a look at those oval things. Are most likely fungus spores, right? What you see over here. Many of the things in the background are probably bacteria as well. But you know what I'm gonna do now is just for the sake of it. Yeah, I'm gonna go back. This is 40 times, right? So let's try phase contrast. I have only one phase contrast objective, unfortunately. But let's have a look. Let's have a look here. Yeah, and here we actually see it a little bit better. Yeah. Phase contrast is good, especially if you want to look at thin objects as well. Yeah. So, yeah, I don't know if it's contracting again or not. I'm gonna tap now the cover glass. Careful, we might lose it out of the field of view, however. No. That does not seem to cause it to contract. It must be something else. Okay. Yeah. I mean, I hope you get the idea a little bit. So that's kind of but I want to also have a look at the moss. Again, I did not, ever since I made it several weeks ago, I did not look at the moss. Yeah. So to a certain extent, all of this is also a little bit how do you say this? Unexpected for me. It's a little bit also improvised for me because I don't know what to expect either. So I'm just basically on exploring just together with you and seeing it sometimes also the first time. But honestly, this sample appears interestingly a little bit less alive than I was expecting. So, but let's move over here. This is now with a cover glass where there is no cover glass. So there is now a little bit more movement possible. Yeah. Yeah, but difficult because of the lack of depth of field. Yeah. So let's go up again. Yeah. So that's essentially decomposing wood, and here's something. I personally am a little bit surprised that those single-celled organisms that are kind of swimming around like this, that on a moist piece of wood, that there is apparently still enough water for them to actually move around. So. I mean, for the worms, they're fairly strong anyway, so no surprise there. But for those single-celled organisms, actually, they need to swim in water. So apparently this very thin layer of water on the wood is enough for them to survive. Yeah. Cold window temperature could be. Yeah. Yeah, again, there was the question every now and then, where do the colors come from? I have a so-called a DIC microscope, which allows me, it uses polarized light and some fancy filters, and it can do all sorts of color plays, and adjust the colors like this. Yeah, in natural colors, it would look, it's much brighter. So this is actually the regular bright field. So if you have got a microscope at home, most likely you're going to see it like that. So These are the natural colors of the specimen. Yeah, and sometimes, yeah, what you use that basically wherever you see it better. So yeah, so let's have a look at a little bit of moss. Okay, occasionally I'm checking again always the time, and this is now where honestly where we have to be. How shall I say this? If you're not able, general suggestion, if you're not able to find anything interesting in moss, change the moss sample. Instead of just trying to look for more longer, generally change the moss sample. When I got the moss a few weeks ago, it was, I think, one of the best moss samples I ever had. There were so many tardigrades and other critters in there, it was unbelievable. And I do not know whether they actually have survived that long. Okay? Ooh, did you just see that? There was a little insect which came out. Huh? So this is a little bit a question. I don't know, how well they have survived. So we're gonna see. I really don't know. So I'm going to remove. First of all, I'm gonna remove this piece of and I'm gonna take some of the moss out. You see, there is some. Let's take this out here. And you turn it around like this because of the soil and everything you want to have on the top. And then you do the following. You add water. And you do it like this. And that's enough. We put it back. Yeah, I'll make it, I'll just drop it in like this, but I'll make it nicer later. All of the big things that we've got in here, I'm just going to remove. Then you do the following. You go like this, and you add here's something here on the back. And you wait a minute or so until everything settles down. Yeah. Because, many microorganisms or microanimals are denser than water and they will settle down. Then you do the following. You take a pipette and, which one? Where did I put it? Where did it, okay. I've got a whole bunch of disposable pipettes, but I don't want to always use a new one. Look, those I cut off the tip because I use those to catch water fleas, which are a little bit larger. So what I'll do now is I'll simply use one of those that I have. So I need to open this. I can't open this. Here we go. Yeah, I also, after using them, I also rinse them. Of course, I have got also microliter pipettes like these, but the problem is with those, I have to cut the tip a little bit because they would get clogged by the particles that we've got here. So, and now we do the following again. Slide here. I'm just gonna go in here and I'm gonna take some of these things up. Maybe a little too much, and then you wait. You wait again for the things to kind of settle down on the bottom. Yeah, all of the denser particles and microbes and whatever. Maybe not the single-celled ones because they're floating around a lot, but slightly larger ones might actually settle down on the bottom. Can we actually see this better? You can see how it starts to collect. Okay, let's wait another minute or so. Yeah. So, and I'm gonna just use this time also to go through the chat. Yeah. Could the fact that the jar was closed have an impact on the small number of microorganisms, or does the moss provide enough oxygen? Honestly, yes. And I did not keep it closed all the time. I have to be honest with you. And I would say the plants that are in there, the moss, for example, yeah, during night when it does the respiration, it will actually use also oxygen. So I would say that yeah. So generally, I think it's not a problem. I just want to show you something else here. The jar with this is a jar with vinegar eels, nematode worms. They move around vigorously, yeah, and I keep it completely closed all the time, yeah. So the oxygen that's in here is enough. Or over here, I've got some water fleas. I don't know if you're able to see them move around. Yeah. I keep the jar closed all the time. I only open it every now and then when I feed them. Yeah, but the oxygen is enough. So, yeah, I think this might be already enough here. Let's try it. Okay. No idea if this is gonna work or not. I'm just going to maybe just push out the rest here. Cover glass goes on top. And we are ready to observe again. Let's see if I've got a single one. Okay. This is a typical example if there was something too big in here, right? We're just gonna leave it as it is, okay? Yeah, so maybe we're not able to use the highest magnification because it's too thick, but we're just gonna have a look at what we have got here. So, again, we're going to and the scope view, and there's something moving already. There's plenty of stuff moving. Let's focus again. Usually it's a better idea to get some overview at a low magnification. Worms. Yeah, so what do we learn? There seem to be more worms in the moss than in the decomposing wood. Yeah. Oh yeah. Is this some kind of a mite? Or what is this? Okay, let's have a closer look. Let's have a closer look what this is. Looks like a microscopic mite. So arthropods are also present. Okay. Yeah. I'd have to count the number of legs, but usually it's nothing uncommon to find microscopic mites as well. Mites have 8 legs. This doesn't, but the body shape doesn't look like one. I don't know. Yeah, so, yeah, you see that in, of course, different parts of the sample there are different organisms. Why am I showing this? Because occasionally people say, "Oh, we don't have access to a water sample, and I want to keep something to observe." So that's one possibility. I've got my jars standing around, and also those here. And if you've got, because this was also an email that I got recently, I'll make a video on that as well. Questions about parents and grandparents who want to do some microscopy with their kids. Right, and you want to always have some kind of samples to quickly look at. So I recommend making this kind of mini terrarium, maybe, yeah, simply to keep it. And then when you want to look at something, you can go on exploration, so to say, armchair exploration. Yeah. So, there. Yeah, last time when I looked at this. I was able to find tons of, maybe if you remember, just check a previous live stream, I was able to find tons of tardigrades. And this time Not so many, but for that different, yeah, like worms and so on. What is this here? This is a huge thing here. I don't know. So I'm going to quickly read again then some of the comments. Yeah, and plenty of single-celled organisms as well. This kind of also shows that there is a so-called a progression. So if you, of course, put it into a tiny jar with a lid on top and into your living room, or yeah, in my case, in my work space, temperatures are different, light conditions are different, everything's different. Of course, there's a progression of different microorganisms. So certain microorganisms start to disappear, others increase. That is a normal progression because the environmental conditions are obviously not the same. Yeah. And if you change the environmental conditions, then you're kind of favoring the growth of certain organisms and not others. Yeah. For example, these worms here are quite nice to look at. Just a second. I've seen something. Do you see this thing in the middle? Could be an amoeba. Yep, it is. Good that we found one. Because they're difficult. I mean, they're not there are plenty of them, but they're sometimes difficult to catch because they don't move very quickly. So let's go up. Here we go. Yes, very nice. Okay. Yeah, so that over here, that's the nematode worm. And so it's a multicellular animal. And this, okay. This thing here that you see crawling around, yeah, that is an amoeba. I mean, honestly, amoeba doesn't say anything. The word amoeba can be so many different things, right? Yeah, it's just more a descriptive term on the way that they move. Yeah. So, yeah. So yeah, you see this. It's also not one of the biggest ones that I've ever seen, but in any case, yeah, you can actually watch them move around, and I think it's kind of, it's kind of interesting and cool to see how many different methods there are in the nature to move for movement, the swimming movement, wiggling, and crawling around. Yeah. Now, look, when I'm focusing right now, I'm using the fine focus. Do you actually see that there is this movement, the wiggling back and forth? And that is because the objective actually touches the cover glass. And it touches the cover glass, and when I focus, then it applies pressure on the cover glass. This is not a big problem. It's not ideal, but it's not a big problem because the microscope objectives have been designed with the lens being deeper in, right? So when I do that, it's actually not the lens itself that's touching the cover glass, yeah, because the lens is actually put further in, and it's actually the side of the objective that's pushing down, yeah. So that's kind of a little bit of a protective, mechanism here, yeah. So let's have a look here. I got the wrong setting for DIC, I just realized. This is the correct DIC setting. You notice that it looks a little bit 3D. Now I can adjust the colors a little bit accordingly. Yeah. And in contrast, no pun intended, I'm now going to remove DIC, a regular bright field. It's too bright, so I have to close it a little bit. Go down with the brightness, and this is now regular bright field. Okay. So yeah, I'm happy that we've also found an amoeba. I have to turn it up again. You see how much light DIC uses up? So I have to open the condenser again. Yep. Huh. It seems to even attach itself to surface here. So let's go down a little bit with the magnification. And let's go for some other observation here. Yeah. So it's of course difficult to see if the magnification is too high. What we've got here. Oh, there are two amoebas. Well, this seems to be a nice place for those. Here in the middle right now over here, you see one. There's this one here, and on the bottom, I move it up here. This here is another one. Over here. Oh, this one is starting to crawl around. Yeah. And the slime molds essentially, they will also have individual cells that move around like that, and then they will join up into a larger structure that will then kind of spread over the petri dish. So what's the conclusion? Obviously, the little microterrarium evidently does work. This long thing that's most certainly is some kind of a bacterium that we've got here. Either one of them or many of them, joined together. So I'm going to quickly read again in some of the I'll be interested in Oliver's answer. I don't know the question. Okay, does your stereoscope magnify enough to see the amoeba in 3D? Definitely not, unfortunately. I can show you. Thank you for some questions. I got to get into the questions here. So maybe that's actually a good point, because sometimes I do get questions about stereo microscope to be used with, are we able to see those cells? And I tell you right now, nope. Stereo microscopes are not designed to do that. Okay, but I suppose I have to find the original question here. Is there a physical limitation to the size of a cell? A chicken egg is, as far as I know, a single cell. This is that's a complex question. How shall I don't even know where to start. This is actually a question that I'm teaching in school. It's part of the syllabus. Per definition, a chicken egg is seen as a cell. However, you see, if you look at muscles, for example, like my muscle, I bumped my microscope. Muscle fibers can be up to 30 centimeters long. Essentially, it's one cell containing hundreds of nuclei, right? One of the longest nerve cells in the body can be a meter long. The axon of the nerve cell is 1 meter long, right? Very thin, of course, but 1 meter long. Yeah. So you see, cells can be extremely diverse. Yeah. There are those certain green algae called acetobularia. I don't even know how to spell it. Acetobularia, which are about 10 centimeters tall and basically have one nucleus. So per definition, it's one cell. So what you see is there is even among the cells a huge how do you say, range of sizes, the whole thing why cells generally are so small is because of the surface area to volume ratio. But if the structure is larger, then there have to be other ways how this lack of surface area can be resolved. That we're going pretty into deep biology here. But to give you your short answer here is that there is a huge range of cell sizes. Yeah, so you'll find And then you can debate, is a muscle cell that's 30 centimeters long, should this be considered one cell, right? Or is it just several cells fused together? Yeah, so is a slime mold which contains thousands of nuclei, but actually enclosed by one membrane. Per definition, could be actually seen as one cell. Yeah, so it's also a question of definition. Yeah. Oh no, I did not. Yeah, I did not have bioluminescent dinoflagellates, unfortunately. Yeah. So what I'm gonna do is those are cool jars. Are you referring to this jar over here? You know where I got this jar? There were fruits, peaches were in here. It's actually I bought it in the supermarket. I ate the fruits and yeah, I kind of used this here. So what I'm gonna do now very quickly is as we're kind of looking more or less at yeah, not so interesting at some of the things that we've got here. I want to go through a couple of the questions that you sent me as well. Where are they? Where's the desk again? Because usually at the end we usually talk about some of the questions. From what I found online, maybe the information is wrong, but some seem to be fungi and others aren't. Slime molds seems to be a blanket term. Yeah, for many organisms that come together to create a network. The term slime mold is basically refers to how do you call this? Is simply a term based on the way that they behave. Right, it looks slimy and they move along, but it can be essentially polyphyletic, as we would say. Yeah, maybe two slime molds are not even related. That could quite well be, because simply they have maybe evolved the same way of movement, maybe separately. So this is can be indeed a blanket term. Yeah. Did you ever look at the microscopic needles of the surface of stinging net? Yes, I did. I made a video, and they look pretty nice. A very nice example for stereo microscopy. Okay. And then later, if I don't forget, I'll show you how this whole thing looks like under the stereo microscope as well. Would you be able to update the items on your Amazon? Yes. Oh, okay. Yes, I'll do that. Okay. I know I have this affiliate page, and sometimes they exchange the products, and then they're not current anymore. Yeah. My 24/7 cam. I actually once tried to do that. I will tell you what the problem was. Before I started live streaming a couple of years ago, I actually set up a microscope and I tried a practice live stream for a few hours simply by putting a microscope slide with some worms. Yeah, and simply 24/7. The problem were actually two problems. Sometimes there was nothing to see because the worms moved out of the field of view. And the other problem was that the water on the slide started to evaporate. So what I'd have to do to have a 24/7 cam, I'd have to make sure somehow that the environment is sufficiently stable that I'm able to actually do a live stream like this. And having actually saying that, oh my gosh, just realized I do have something that would make it possible. Maybe I'll give it a try. I don't know if it's a good idea. I don't know if people are going to watch if I'm not talking. Maybe some kind of music in the background. I don't know. But look what I got here. Some of you have already seen this. This is a mini aquarium. Yeah. So you can put a sample in here, and there's the organisms are able to swim in here. So there is plenty of water in here, so you're able to keep it without actually having evaporation. Yeah? So that is actually something I might want to try. 24/7. I don't know, some worms maybe, because worms move around a lot. Yeah. I'll see, but I have to admit, I doubt that people would be very interested in that without any narration or anything like that. Yeah. Idea for a future video: looking at a piece of your fingernail cuticle, since it's such a thin layer of cells. Shall I be honest with you? Shall I be very honest with you? No cells. Fingernails, just like hair, is made of a protein called keratin, not like the carrots. It's it's spelled a little bit different, and so there are actually no cells in here. might still be interesting. Maybe I'm just going to try it as well. Okay, different brands of toothpaste under the microscope. It would have been, yes. They used to put into some toothpaste, they used to put, how do you call this, diatomaceous earth some time ago. This is like silica. To use as a fine powder, as an to help clean. I don't know if this is very good for your teeth because it might be like some kind of very fine sandpaper. But this is at least something that I read, that some toothpastes actually used to have this diatomaceous earth mixed into them as a form of to help clean the teeth. Yeah, and those diatomaceous, then you would have been able to see diatoms in the toothpaste. I recall in one of your videos mentioning the material to capture snowflake impressions. Do you recall what it was? It's snow here. Yes, I can talk about this. Okay, and I made a video, not in this channel, but in the main Microbehunter channel. So what you do is the following. If you want to catch the impressions of snowflakes, you need a little bit of patience, but it might actually work. What you do is you take a clear nail polish and you freeze it in your refrigerator. Really make it really cold, and it only works if it's also pretty cold outside. And what you do is you then cover a microscope slide with a layer of clear nail polish. I mean, I don't have clear nail polish with me right now. What you do is you put a drop you take the brush and you take another slide and you simply smear the clear nail polish across the slide, and everything freezing cold. Right? Also, the slides have to be freezing cold, and you go out and you allow snowflakes to fall on the clear nail polish, and then you keep the slide out there until in the freezing cold, until the nail polish has hardened. Okay, that's it. And then but this only really works if it's really cold, because otherwise what's going to happen is that as soon as the snowflake falls on here, it starts to melt and then you don't get anything. But if it's really cold outside, and if the nail polish is cold and the slide is cold and everything's freezing cold, then you can actually capture the impressions of you can make of snowflakes. Yeah, on nail polish. Looks quite nice. Okay, I made a video some time ago, but but you got to be patient with that, because sometimes the snowflakes come in larger flakes, and you want to actually have the individual crystals. Try it out. Make 10 slides, 20 slides, put them out in the snow, catch some snow, and yeah. Or if you can't catch them, because it might be too difficult to run around with a slide trying to catch snowflakes, what you do is you take, just as a quick trial, you take some loose snow, sprinkle it over it, and that's it. Yeah, that's then at least you can test if it works. Yeah. I wonder if you could cross a section of a human hair with a microtome. That's difficult. I tried. No. I will tell you now an easy way how to get a cross section of human hair. I've done it before, not with a microtome. What you do is the following: you shave yourself. Seriously, this actually works. You shave yourself and take the foam and rinse it with some water and put some of that stuff. Maybe that's an idea for next week. Yeah, it actually does work. You don't need a microtome. Just take a razor and shave yourself, and you'll see the cross section of human hair. Yeah, Moss looks like it's doing well. Have you ever seen anything completely new, like that no one has ever seen before? I will tell you the following. Yes, every time. This is a biology answer now. Okay, have I seen something completely new? I'll show you. I know you're going to be a little bit disappointed what I'm going to tell you right now. Some microbes. You know what? Let's turn on phase contrast. Turn up the light down here. Let's look at something completely new. Do you see those dots in the background here? Yeah, so look, some of these are moving around here. Could be flagellates, or some of these dots are bacteria. Many of these bacteria, most of them, as a matter of fact, are completely new. Nobody's seen them before. You might even not be able to grow them, yeah, because nature is so diverse that essentially many of the things that you see here before, are completely new. Yeah, have not been described yet. Are new species, new genera. Yeah. So I'm just saying, this is, however they look similar on the outside, but actually genetically they might be quite different. Yeah, and might be species that have not been described yet. As a matter of fact, it's not even difficult on that level to find new species. Because most of them have not been described yet. Finding new animal and plant species, especially when not animal, it's still too general. New mammals, for example, that's apparently pretty difficult. Yeah. So the higher animals, they've already been found, right? But concerning the microorganisms, most of them that you see here have not been described yet. Yeah, and probably never will be, because you cannot culture them in the lab. Yeah, so this is an interesting question. Concerning something completely new. Could the fact that the jar was closed have I talked about this. Not likely. Cool jars. Have you ever? No, not done any dinoflagellates yet. Take a sample from the finger of your granddaughter and watch it on the microscope they made, so the fingers into their mouth. Shall I be honest with you? Yes, I get what you mean. Fingers are dirty, no question. You know where you're going to find more bacteria? In your mouth. Because actually the mouth is full, it's a moist place, it's a warm place. You've got food there, right? I mean, these are not bacteria that are normally, of course, growing in your mouth. So our body's immune system is able to deal with that. And of course, fingers are horribly dirty as well, especially if you, for example, you can spread diseases this way, obviously, and then you put them into your mouth. However, no question that there are way more bacteria, at least in your mouth, than probably on your fingers. Yeah, so that's actually quite interesting. The human body, luckily, is able to deal with that. Yeah. If you discovered an organism or a genus, are you allowed to officially name it? Yes, you are. You are. And as a matter of fact, you can propose a name, and there is only one condition. You're not allowed to name the organism according to yourself. Okay, so that is, you can name it according to another researcher, another important person. You can, the names are Latinized. So essentially they sound Latin. Yeah. So those bacterial names like Escherichia coli, Escherick was a scientist. Salmonella, you've heard. Mr. Salmon was an American veterinary doctor back in the 19th century. So you basically name bacteria. Brucellosis, the bacterium causing the Black Plague, Yersinia pestis. Yersin, he was a bacteriologist. So many of those organisms which have these fancy names actually are named according to people. Yeah. So this is simply something that I just want to tell you. So it's also something that I tell my students when they read some kind of text and they have this long, complex names. Yeah, and it says, just somebody invented those, right? It's nothing complicated. Somebody just had to come up with a name, right? So in that sense, it's you are allowed to name them. Yeah. Let me quickly go through some of the points again here. Okay. Yeah, I love the mini aquarium. Yeah, by the way, mini aquarium, I just show you, because some of you, I mean, many of you already know this because you've been in the live streams before. I'm just going to show you what my mini aquarium is. It doesn't look nice, but here it is. Many of you remember this. Look, not nice looking. The uglier it looks, the more interesting it is. Okay, there's stuff floating on this. Is this some kind of fungus growing here? I have no idea. Yeah, but this is basically a jar for cucumbers. pickles. I think, yeah, yeah, and yeah, I simply collected some water and put some rocks in here. No fish, and yeah, you see it already evaporated quite a bit. No, this is not possible. Come on, here we go. It already evaporated quite a bit. So I have to add more water. Remember several months ago, I cleaned out all of my jars and I simply dumped everything into it. So it was kind of a mix of different, you know, types of water. So this is also a possibility, and yeah, you can actually see there are lots of algae in here. And every now and then I fill it up again with water. Yeah. So this is kind of like an aquarium under quotation marks. Let me put it back. So here we go again. Huh? Oh yeah, by the way, this is how my, oops, I see this pretty, lots of dust. But this is, in case you're kind of wondering, this is how my whole work space looks like. I'm just also going to show this to you a little bit because, yeah, in a couple of minutes, I'd like to call it, quits as well. So what I've got is the following, of course. Look, this is, yeah, my monitor. Yeah, so where I'm able to actually see everything that I'm doing on screen. Yeah. So, and down here, this is where I've got all my tools, and then of course the table. Yeah, and on the side, I've got over here next to the window. Yeah, microphone is here. It actually looks pretty much messier than it actually is. Yeah, so this is how it looks like. Don't miss my last. So has there been a question somewhere? Okay. Yeah. I don't know. I can't. I hope I didn't look over, forget about any questions. Okay. Yeah. Yeah, it's not an entire lab, honestly. It's just a larger table that I have where I simply keep all of my stuff. Yeah, and I've got my main microscope and my stereo microscope next to me where I cannot swing the camera over. Yeah, so here I resend it. Thank you, because I could not find your question or your comment. And but that is something. Thank you for the recommendation. I will maybe, I don't know if this is maybe I'll try this over the Christmas holidays, maybe in a few weeks, when essentially I am at home the whole day during the holidays, or maybe on a weekend once, because I don't want to keep the computer running and the microscopes running when I'm at work. Yeah. So, but this is actually indeed something I can put something in here. And then maybe yeah, have a non-stop 24/7 live stream. Yeah. Okay, I still you know what I'm gonna do? I've got top chat. Maybe I click on live chat with the correct so I switched over to live chat. There's a question here. Have you finished your vacuum chamber yet? Not, unfortunately, not yet. I bought a vacuum chamber. But it did not contain, it was such a cheap one. It was a manufacturing problem. It did not contain a hole to contain connect a vacuum tube. No, not vacuum tube, a vacuum hose to it. Yeah. Yeah. Let's see. Really? Okay. Yeah, so that is, so you, ever smaller, you seem to be having some problems posting something. I have something under my nose. So I think, what's is it? Zit is a little, how do you call this? Infection, you mean? I never heard of the term zit before. British English? Yeah, I mean, I don't know. Do I have one? This full of white blood cells. I already did this once. Seriously. Yeah. Yeah, I don't know. This is not gonna work well. because simply not enough white blood cells. Nah, it's not gonna work. Not enough. Unfortunately. Yeah, but I've actually done this once. You squeeze it out, you put it on here, you stain it with methylene blue, and you're able to see hundreds of white blood cells. Yeah. So it's also something to look at. Yeah. Yeah. A pimple. Ah, I see. But I can't. You see, I need a mirror to actually squeeze it out. Yeah. So, but actually, I have done this already before. You might actually find it either in this channel or in the other one. And what I've done is if you get those and squeeze them out, you have to really make sure that because the cells, the white blood cells, are so dense together, you have to separate them. You really have to spread them out a little bit. Okay. I don't know. Yeah, honestly, if I have one, but I have to prepare it. But I kind of wonder. Yeah, I don't think that I got a lot here because I already scratched it open. But maybe we're lucky. I don't know. Let me have a look. I have doubts. But you always need to test it first and see if it. Yeah. Let me see. And yeah, it's plenty of other stuff. I probably scratched off a lot of my skin cells as well. Yeah. Let me see. You see, I don't even. Yeah, lots of dirt and stuff. Probably some dead skin cells. Some kind of fibers. But honestly, not. Yeah, this has to be a more cleaner, under quotation marks, sample than what I've just collected here. So it is probably not going to be. This is probably not the best one. Look, all of those things that you see are definitely skin cells that have flaked off. Yeah. Because those skin cells, they come off quite easily, and those white blood cells, they actually look quite interesting, especially when you stain them, because they have this irregularly shaped nucleus. Yeah? So, as a matter of fact, skin is one of a huge component of house dust. Actually, is made of skin. Yeah, those dust mites love to eat this stuff. But now at least we've also looked at some skin cells under the microscope. Okay, so let's have a look at some of the other comments here. Yeah, otherwise it's already 1.5 hours. Yeah, so, and what I'm going to do therefore, I'm just quickly scanning through the comments. Now I also learned a new term. I did not know the term zit. A mini aquarium. Nope. Yeah, I think I got pretty much all of the questions covered, more or less. Yeah. An introduction to digital adapters and cameras, etc., for microscopes would be cool. Yeah, actually, if you're interested in that, go in this very channel. A couple of weeks ago, I actually did talk about mobile phone adapters and connecting a DSLR camera to a microscope, and I've showed you some of the equipment that I have. In this very channel, just if you go and click the live stream tab, all of the live streams are actually can be still watched, and then you can have a look at some of the adapters that basically I've collected over the years. What I'm going to do now is now, I didn't expect that we're also going to look at my skin cells, but see, that's the nice thing about live streams. If you give me recommendations, I will try to look at those things. But from my side, I think I'm just going to call it a day again. Thank you for having joined me again, and I will be making again, just a organizational how do you say, information. In case you're kind of wondering, I'm now of course continuing the live streams on this channel. There is the other channel called Microbehunter. This one is called Microbehunter Microscopy. The other channel, Microbehunter, I'm going to do more explainer types videos about biology. So it's going to be less microscopy focused, but it's going to be more, a little bit more theory and other things. Yeah. So just for your info, if you don't know, but there are two channels. I've got another channel as well, and I'm going to call it a day today because I already feel that I'm starting to become a little bit sore on my throat, and I think I'm just going to, yeah, wish you all the best. Hope that I'll be able to see you again next week. Happy microbe hunting. And yeah, and all the best. Bye bye.